Hello, everybody. I'm Milton Korn of the National Library of Medicine. I welcome everybody to the inaugural lecture of the Lindberg-King Lecture Series. Just a word about the series. It's endowed for this year. It was endowed this year. This is the first lecture in the series. It's intended to, each year, have a lecturer here who will bring us something useful and important to NIH in the field of biomedical informatics or pathobiology, and it was created in order to honor two distinguished scientific leaders, Donald Lindberg and Donald King, both of whom have had a long association with the National Library of Medicine and with the National Institutes of Health. I'm delighted to say both of them are here, and after Dr. Altman's talk, they will present Dr. Altman with an award. This lectureship was endowed as a co-sponsored achievement with three organizations: the National Library of Medicine, the Friends of the National Library of Medicine, and the American Medical Informatics Association. And I would like to have the leaders of these respective organizations step up here just to make some brief remarks about it. Betsy Humphreys is acting director of the National Library of Medicine. Could you come up here, Betsy?
Well, the National Library of Medicine is very pleased to co-sponsor this lecture series with the Friends of the National Library of Medicine and the American Medical Informatics Association. All three organizations have benefited greatly from the leadership of the two persons for whom the lecture series is named. I personally have benefited from the ability to work with and learn from both of them. I think that this opening lecture is a great start to what I assume will be a very great series and contributing to the goals and the missions of all three organizations. So I welcome you here today, too.
Thanks, Betsy. I might add that this talk is being videocast widely, both on the campus and outside of the campus, and it will be archived as well. We also have here Glenn Campbell, who's president of the Friends of the National Library of Medicine. Glenn?
Thank you, and welcome, Dr. Altman. It's a pleasure to meet you and a pleasure to have you here. We look forward to the lecture today. I also just want to add my thanks. It is a pleasure to be a co-sponsor with the National Library of Medicine and with the American Medical Informatics Association on this distinguished lecture series in honor of these two very great men whose contributions are well known to all of you. We're also very grateful for the endowment that is funding this inaugural lecture and will fund the subsequent lectures in medical informatics and in pathobiology. So welcome, everybody, and we look forward to hearing you, Dr. Altman. Thank you.
And lastly, we're glad to have here Doug Fridsma, the president of the American Medical Informatics Association.
Thank you. I will join my colleagues in just being grateful that this lecture series has come together to honor both Dr. Lindberg and Dr. King. I'm also delighted, too, that this inaugural lecture goes to a good friend of mine and someone who has been a long-standing member of the informatics community, Dr. Altman. And so I look forward to sharing the insights that he can provide to us and kick this off, I think, in a really great way. So thank you.
Well, our speaker, Russ Altman, what to say? We have a abbreviated bio in the program, and I wish you would take a look at it. But I think above and beyond some of the details in there, I think he represents something that's new, of increasing importance, and its importance has only begun to blossom. And I'm talking about the application of computing to molecular biology problems. In other words, bioinformatics. There was no bioinformatics to speak of a few decades ago, and now I think for many people it's one of the really important fields for making advances in biology and in medicine. And Russ is one of the pioneers in that. We wanted to start this lecture with someone who was internationally distinguished in this field, and he's the man we wanted, and he's the person that we got for this. He is also one of the very early graduates of one of the NLM's programs in biomedical informatics many years ago at Stanford. He stayed at Stanford, and now, as far as I can see, he owns Stanford. He is, I believe, a member of 3 departments there, and while we didn't have a chance to put it in the program, he's going to be a member of a 4th department as well. And Russ, if that means you're being paid by 4 departments, we like this even better. In any case, it's a pleasure to start this off with somebody with this reputation and with this record of achievement. Russ?
Thanks so much, Milt, and thanks to all of the people who helped put together this lectureship. Congratulations to Dr. Lindberg and Dr. King. Yes, who I'll speak about in a moment briefly, but yes, you can see my departments. This is listed in the order of how much they pay me. It doesn't mean multiple salaries, it just means multiple slices of the same salary. And the new one is the Department of Biomedical Data Science. I'm sorry, the font's got a little bit revised here, but it's a brand new department, started October 1st at Stanford. It will house our biomedical informatics training program, which I direct, and we're very excited about it. And I do not want to leave till the end the acknowledgement of the work, because I think particularly in light of Don Lindberg's presence here today, I want to point out very clearly that the work that you're going to be hearing today was supported in part by a grant from the National Library of Medicine that I got in 1994 and is in its 21st year, 05652 for those of you who are taking notes. And we are incredibly grateful for that support. It has allowed us to make contributions in a couple of areas, and also NIGMS has supported the work for quite a while as well. And so we're very grateful to all of NIH, NLM, and NIGMS in particular. This title is a little bit of a tongue-in-cheek, because you could probably call almost anything could be this title, Integrating Multiscale Data for Biomedical Discovery and Clinical Information. The reason it's tongue-in-cheek is I'm going to be talking about natural language processing. This is partly a lecture honoring the legacy of the National Library of Medicine and Don's leadership of it, and Dr. King's participation. And the best multiscale data in the world is the biomedical literature, because it talks about atoms, molecules, tissues, organisms, populations, the environment. So that's the multiscale. And the question is, we have this amazing collection of biomedical text. How do we use it for discovery and for helping patients? And I want to give a flavor of some of the work we're doing in that area today. So there's the grant. First, I just want to say a couple of words about Don. As you know, he stepped down as director of the NLM recently. He had been the director of the NLM for my entire professional life in informatics. I did not know an NLM that was not directed by Don Lindberg. In 1984 January, many of you who are old enough will remember that there was a Super Bowl commercial that announced the Apple Macintosh. I remember making a pilgrimage at Stanford campus to look. They had one on display, and it was a little bit like Eva Peron's funeral, where we were allowed to walk by it and look at it just for a few moments and then decide if we wanted to purchase it. This changed my life, and I won't go into too much detail, but it literally made me change my program from biophysics to a program at Stanford called Medical Information Sciences in the spring of '84, around the time Don was beginning to lead the NLM. As I mentioned, NLM granted me my first funding in 1994. They added a Presidential Early Career Award, for which I was very grateful. And really, this is to the entire NLM community, but in particular to Don, I'm very grateful for your support of the field and your support of my career. This is Don's word cloud. I had the privilege of serving on a committee that was writing, with actually Doug Fritzman and others, that was writing a future vision for NLM in the post-Lindbergh era. What are the opportunities? What has to happen? And one of the things we did is get public commentary, and I can tell you that never was there a clearer signal of a NIH institute being beloved, and arguably the most beloved international NIH institute. And there are some very strong institutes that deserve love. But I think because of PubMed, so this is a word cloud of all of those responses from the community when asked about the future of the NLM. And the bigger words were mentioned more often, and I'm sure you will find some of your favorite NLM activities, the WISER system, chemical information systems, health, PubMed, et cetera, et cetera. But what was clear is that as an outward-facing resource for the world, the NLM is almost an emissary to the international community, which is an embodiment of NIH's mission to help global health. And that became very clear, and I think almost all these words are there because of Don's leadership. So I call it Don's words cloud. Okay. So my lab is interested in drug response, and in particular, the genetic and molecular basis for drug response. How do drugs work? It's kind of a miracle that they do work at all. They don't always work. How do they cause toxicity? How can we design or adopt new drugs or new drug combinations to more efficaciously treat patients while minimizing side effect? And part of the way we do this is we're building this database, knowledge base called the PharmGKB, the Pharmacogenomics Knowledge Base, which is the repository, we like to believe, of all information about human genetic variation and how it impacts drug response. We know that there's a genetic basis to drug response. You inherit that from your parents and grandparents, just like you inherit their eye color and their hair color. But unfortunately, we don't have family lore about that. And we don't sit around the fireplace saying, "Oh, Grandma, she had a heck of a time with that warfarin." I mean, unless it killed her, then they might talk about it. But otherwise, you don't have that. And so we're forced to measure DNA in order to understand the genetic influences on drug response. And we've been building this resource for about 15 or 16 years. Please visit it because we get credit for hits. So, and they don't count if they come from the Stanford IP address. So please go to a non-Stanford IP computer and search for PharmGKB. PharmGKB is about understanding drug response, both the pharmacodynamics, how a drug works, and the pharmacokinetics, how a drug is metabolized. Here you see for the drug warfarin, which is the purple box at the top. You can't read it, but that's okay. You see a series of genes that are involved in how warfarin works. It's pharmacodynamics, and obviously it's an anticoagulant, and in the end, you're more likely to bleed, less likely to clot. This is built by our team. Every ball on this graph and every arrow is supported by literature in PubMed universally, okay? And so it's extremely expensive graph. I have PhD level curators at the PharmGKB, 6 or 7 of them, who are on a daily basis perusing the literature, pulling out important information, and putting it on a graph like a map like this to help people see an integrated view of how a drug works. They do the same thing for how a drug is metabolized. So here you see the metabolism of warfarin, which is an entirely different set of genes. These are mostly cytochrome P450 genes that do oxidoreductive reactions, and through a various set of transformations, render the warfarin inactive. And the vision for pharmacogenomics clinically is that we will measure your genome, put it in a secure database, and then when your physician or healthcare provider or pharmacist is about to write a prescription, they use this system to try to pick the medication that is most likely to be efficacious and least likely to cause side effects. And you'll know which genes to look at because we will have made these pathways. They will be complete, or as complete as we can make them. And so understanding all the variants in those genes, which are the little eggs, the little blue eggs, will be the task at hand. And we will know if it worked because we will see better drug response in terms of efficacy and less toxicity. I apologize for this slide. It's just a page in the website, but it's just to show you that we really go down to the individual base level in this knowledge base. So here you have for warfarin 2 genes, a cytochrome P450-2C9 and a gene called VKORC1, and we have an ID of a SNP. This is a SNP ID that gives a unique location for a single base in the human genome. This is the gene, and it says if mom and dad both gave you an A, then you have an average response to warfarin. If mom and dad both gave you a C, then you're going to be very sensitive to warfarin and need a lower dose. And we have thousands of such summaries in the PharmGKB set up so that the next phase of clinical implementation can happen. All of this extracted from the literature by our curators, reading, making judgments, and putting it into the database. We wrote a paper in 2009 saying, okay, where is pharmacogenomics today? A colleague of mine sequenced his own genome, Steve Quake, challenged a team of us, and you can see it was a pretty big team, to look at his risk for common disease, his risk for rare disease, and his drug response. My team did the drug response analysis, and we found that for hundreds of drugs, we had something clinically relevant to say. I put on my general internist hat, and I said, okay, which of this data actually would make me pause or change my mind about a drug prescribing decision? And there were hundreds of them, and there were a handful of them that I would call very high value. He is very sensitive to warfarin, and if he's ever going to be on warfarin, he would need a low dose, almost certainly. And he would have an unusual response to the drug clopidogrel, which is used universally in cath labs around the time of when people are having angina and potential myocardial infarction. So those right there probably would have made it worth it. But there's a long tail of other advice, often at a lower level of evidence, but often still clinically useful. And again, all of that information came from the literature. So let's think about the literature and this task of getting this information out of text into our database. So you've all seen pictures like this. This is the growth of the biomedical literature only up until 2010, so it's a little bit of an old slide. And the pharmacogenomic literature is having an even quicker kind of uptick, and that continues for the last 5 years. And at the end of the day, what my curators are trying to do is pull out relationships like this. So in the gene CYP2C9 metabolizes warfarin. So it's a semantically typed relationship. Not just CYP2C9 is related to warfarin, but it metabolizes it. Or cystic fibrosis is associated with the CFTR gene. Or itraconazole inhibits another cytochrome P450, CYP3A4. These are the kind of elemental things we'd like to be able to extract out of the literature. One of the problems, as we all know, is you can say the same thing in many ways. And so in a paper a few years ago, we showed a technology that we had prototyped for actually mapping sentences that say the same thing with different words into a standardized representation, an ontology or a terminology, something that NLM is the central resource in the world for doing. So this sentence says the ABCB1 C3435T polymorphism influences methotrexate sensitivity in rheumatoid arthritis patients. A variant C3435T allele of the MDR gene, which by the way is the same gene as ABCB1, but you would need to know that, affects methotrexate tolerability. So in a paper, I won't go through the details, but in that paper we showed that you can parse this sentence to find the nouns and the verbs and the adjectives and the adverbs. You can map them to raw relationships that are expressed in the initial language of the sentence. But then we came up with a way to map these terms into a standardized vocabulary. Very powerful, because now multiple sentences, this is just two, but we could have multiple, are being mapped into a standard vocabulary where we have a series of verbs that have been standardized, we have a series of concepts that have been standardized, and we can now talk about what's being said in these papers, and computers can think about it because we're now giving them normalized data, if you will. I won't say more about it, but that motivates a lot of what we're trying to do. So now I want to tell you three little stories about text processing in the context of pharmacogenomics to show you how this work has evolved. And the first one is, as we all know, PubMed has more than 20 million abstracts, and increasingly there are publicly available full text. We're very excited about that. Most biomedical knowledge is stored in this literature. And so can computers read PubMed abstracts and actually make new inferences by taking pieces of information that are in different publications, putting them together and making a logical case for a new hypothesis or new piece of information. The thing that enables this is remarkable advances by our colleagues in computer science and linguistics in natural language parsing. So you see one of the sentences that I just showed you from top to bottom on the far left. And here you can see that it's been parsed by something called the Stanford parser, not written by our group, written by my colleague Chris Manning at the Computer Science and Linguistics Department. But what he does is he makes a statistical guess about what the nouns and verbs are. And by the way, he trained this on the Wall Street Journal, but it worked remarkably well on PubMed abstracts, which was a surprise, and we were worried we were going to have to retrain it. But it makes a statistical guess about the nouns and the verbs, and then it builds this thing called a dependency graph, which has the main verb at the top and basically has the subject on the left and the object on the right. I'm sorry if I'm bringing back terrible memories of your grammar school or high school sentence diagramming. That probably the more senior of you will remember this. So this is like a parse tree, but it's related to a parse tree. But here you can see that there's the polymorphism is the subject of the sentence, influence is the verb, and then sensitivity to methotrexate with some modifiers is the object. And Chris and his code can very reliably do these parses. And these are very computationally nimble for us. We can then use them to identify all the verbs that are used. And then we can cluster the verbs. I'll show you how we do that in a moment. But this allows us then to say, okay, at the end of the day, there's a polymorphism in a gene that is influencing a drug sensitivity. When we looked at many of these sentences, we found that 80% of the sentences used a very few number of verbs and concepts. And so that's what allowed us to create this controlled vocabulary of variants, verbs, and effects, drug sensitivity. That's a little glimpse as to how we mapped multiple sentences to a standard format. But what was really remarkable was what my student Beth Percha did with this data after doing this. She took 170,000 PubMed abstract sentences that include both a gene and a drug. She parsed them. She did this standard normalization, as I've just kind of hand-waved a little bit, and built this spectacular graph where drugs are green and genes are, genes are red. And each of these little blue lines represents a sentence in PubMed that relates that drug and that gene. So first of all, you get this beautiful picture that I've actually displayed in this auditorium before. And they basically summarize semantically the relationship between a gene and a drug by a certain verb. So just to blow up one segment, here's ABCB1, which is a transporter. It's on the surface of cells, and it transports molecules mostly out of cells. And it's responsible for a lot of cancer drug resistance. If you've heard about the P-glycoprotein or the multi-drug resistant transporter, that's ABCB1 transporting your chemotherapeutic out of the cell before it can do its good work. And then there's verapamil, a calcium channel blocker used for hypertension and other things. And you can see I've labeled a few of these sentences. This sentence said the drug verapamil is metabolized by the gene ABCB1. And in this case, metabolized means transported. Or drug metabolism of verapamil is influenced by a polymorphism in ABCB1. And so on. We get this very rich semantic information about the relationship of this gene and this drug by doing this processing of these sentences. Okay, so what? So Beth had this great idea. If we have a relationship between a drug and a gene, and if we have another relationship between that same gene and another drug, could that suggest a drug-drug interaction mediated by both of these drugs interacting with the same gene? So she took a training set of known drug-drug interactions and said, what are the kinds of genes that occur in the middle, and what are the kinds of connector words? I haven't been discussing them, but as you recall, there were other words in those sentences that we call context. What are the contexts and what are the genes that are suggestive of a potential drug-drug interaction? So she trained a classifier. I'm jumping over a lot of work, but what she came into my office with was a list of 2 drugs and then rank ordered by the likelihood that her algorithm believed that they would be interacting. And none of these pairs were in her training set. Some of them, the ones in color, were entirely novel. So there's a 0 in this column, because if they were known interactions, even though they weren't used in our training, we still had a lexicon of drug-drug interactions that we could see were right. So we were very happy to see lots of these 1s, which means we got them right. But we were intrigued by the 0s, which means none of the gold standards that we had listed these, and yet they were having very high scores. One example is metoprolol and dextromethorphan. So metoprolol is a beta blocker. Dextromethorphan is a cough medicine, in Robitussin. And they are both, well, let me tell you. In 2000, in this journal, Drug Metabolism and Disposition, there was a sentence, please do not try to read it, because it is essentially unreadable, except by our parser, which really you begin to love, because it looks at this text and says, here's what's being said, Russ. Drug is metabolized by 2D6. That I can understand. So thank you, normalization scheme. That was in 2000. A paper in 2005, 5 years later, said in the abstract, again, a little bit of a better sentence, but basically 2D6 metabolizes dextromethorphan. Now, 2D6 is a drug that can be saturated. And so if a patient is taking both dextromethorphan and metoprolol, it's not out of the question that they would not be able to metabolize both of the drugs at the normal level, and some of them, the drugs might accumulate higher levels than expected, and you would see some toxicity. And in fact, in 2011, after this paper was drafted, there was a case report of dextromethorphan, and I won't ask you to read it, but you can see the word metoprolol here, and it says, because it's bound to 2D6, we believe that this patient had myoclonus after dextromethorphan because of a drug-drug interaction with metoprolol. Very satisfying. That's just a story, but the statistics also bore us out that this was a reasonable way to go, and that we were taking one observation from 2000, another observation from 2005 out of 23 million abstracts, putting them together and making a hypothesis. That's pretty exciting, we think. So that's the end of the first story. The second story is I told you that we were able to find words, verbs that mean the same thing in these different sentences, and therefore we could map them back to a common controlled word. So for example, we might have, well, I'll show you some examples, but we might have inhibition or blocking or reducing. All those might mean the same thing, and then you could map them all to a standard term that means inhibition. But how are you going to do that automatically? And it turns out, once again, our computer science friends have given us some amazing power tools. In this case, I want to tell you about this idea of random indexing. And I'll show it in a picture, which I think will be more clear. But each word gets a random vector of 0s and 1s. You just generate it randomly. It is unique to that word, but it's random. Then you look at all of the language to the left and to the right of that word in a certain window size. And you look at all of the random vectors for the words that are surrounding your word of interest, and you add them up. By adding them up, what you're essentially doing is you're getting a new vector that summarizes the context of words surrounding your word of interest. And then you can see what words have similar contexts. Maybe those are synonyms. So Beth, again, Beth is a senior student. She's been very productive, and this talk is a romp through her career. Hi, Beth, if you're watching, and thank you for your help with the slides. Can we automatically learn synonyms? So what does the word polymorphism mean? Here's a bunch of text which has the word polymorphism in it. It's a little bit like Where's Waldo? If you look around, you can see polymorphism all over. It's a little puzzle. In the random indexing, we're going to take all the words that we care about, and we're going to give them a random vector. 0 is white, 1 is blue, -1 is red. So here's gene, diabetic, and patients, just to show you 3 words that I really meant random. There's no relationship of the vector to the word. But then when we see the word polymorphism, we look to the left, we look to the right, we add up all those words, and we get a new vector. Then we can use good old math to get the vector distance between word 1, like mutation, and word 2, like polymorphism. And if the angle between them is small, we can say, wow, they look like they might be used in very similar contexts and might be synonyms. So we did this for about 500,000 sentences, again, in Medline. And just to give you a flavor, again, this is published, but here's some high-ranking synonyms, which basically passed the smell test. I hope you agree. Inhibition and suppression, very close. Downregulation, upregulation. Maybe that's a false positive. They're not synonyms, they're antonyms. And we actually have been looking at that problem. Incidence, prevalence, very interesting. If you don't know what you're talking about, they're the same. If you know what you're talking about, they're different. Assessment, evaluation, etc. I'll let you look through this. And then we also had verbs, investigate, examine. This was the part we really needed for mapping those verbs to a common terminology. So this is just a little to give you a flavor of how these emerging technologies for text processing that are very statistical, very latent. This is unlike the parsing where we have a very clear model of the roles that the words are playing and how to interpret them. This is on the other end of the spectrum where it's a statistical summary that's giving us useful information but is not giving us precise semantics. Okay, and then the last story is a little bit of a longer story, but I hope you'll enjoy it. As I said, we've gone up by 200,000 since 5 slides ago. We now have 22 million PubMed abstracts. And as I've kind of hopefully convinced you, these contain a goldmine of relationships between gene and drugs. And in fact, every type of relationship that you can imagine between a gene and a drug is probably expressed in the biomedical literature. And so in this next piece of work, we tried to go for the brass ring, if you will, and say, we want to understand not only the detailed interactions between one gene and one drug, we want to know what's the whole space of possible relationships between genes and drugs. And if that's not clear, it's okay. It'll become clear in a minute. And again, these advances in statistical natural language processing allow us to do this. And this is a paper that came out a couple of months ago, learning the structure of biomedical relationships from unstructured text. So here is another one of those dependency graph parses. So we've put it through the Stanford parser. CYP3A4 mRNA expression was significantly increased by rifampicin exposure in human hepatocytes. And that gives you this, I don't know if you consider it lovely or ridiculously confusing, computational representation of that sentence. With everything, I should point out, labeled. We have a noun, we have an apositional modifier, we have some prepositional phrases, we have a verb at the top as usual. So lots of information there. So the first thing we do is we say, okay, this is expressing a relationship of some kind between a gene, 3A4, and a drug, rifampicin. So let's prune away everything except the direct relationship between the 3A4 and the rifampicin. So the next is exactly the subset of this dependency graph that is on the direct line between 3A4 and rifampicin. And basically it says 3A4 expression is increased by exposure to rifampicin. Remember that other sentence that was very long and I said don't read it, and then I showed you this beautiful summary? That's how we do that. We get rid of all the modifiers. They may be important, especially not. We pay special attention to the word not because that means the opposite. But this gives us a single sentence template that relates a gene to a drug. And so one question you can ask is, how many such templates are there, and are they similar? So let me just go through this in a little bit of detail because I think it's a fun idea. Here we have some sentence fragments, atenolol, a beta-2 adrenergic receptor inhibitor, which would get parsed as appositional modifier inhibitor AMOD, which I forget what it stands for. And then we have a very similar sentence also in the corpus. Atenolol inhibits beta-2 AR. Same exact sense, but a different dependency graph path. Now we see lisinopril and ACE, angiotensin converting enzyme. And what do you know? It has a slightly different, well, actually it has the same exact dependency path as sentence 2. Those are identical. These are homologs, except we've swapped out the drug and the gene name. So there's a path that has actually occurred twice in our little tiny corpus that you're looking at. And then we have other versions. Lisinopril is an inhibitor of ACE. Atenolol is a potent beta-2 adrenergic inhibitor. So we get a list of all of these sentences. And that begins to, there's something profound here. I might say that even though I've never seen this sentence, lisinopril is a potent ACE inhibitor. I've never seen that sentence. I might say that sentence could be written because there's so much similarity in the kinds of sentences that are written about atenolol and B2AR and lisinopril and ACE that I might actually speculatively impute that that sentence could exist even though I've never observed it. So I think that's an amazing idea, that based on the similarity of the global use of these genes and drugs, you start being able to write sentences that have never been written but you believe are true. I'll let you decide how profound you think that is. Now, you can go on, and now you say, well, atenolol binds B2AR and lisinopril binds ACE. So you say, wow, maybe whenever you're an inhibitor, you are also a binder. That's interesting. But now we have a new example. Dobutamine binds beta-1, but it does not inhibit beta-1. So that tells a computer that's paying attention that these are synonymous. They are related to the binding relationship, but they are not strictly synonymous because there are examples where you have a negation of the relationship, binds or not binds. And so you can't just say that if I'm an inhibitor, I'm also a binder. So it's this kind of transfer of information between the sentences in the corpus that we tried to automate. And I want to tell you kind of how we did this. Oh, and by the way, these sentences, I'm happy to report, really do appear hundreds or thousands of times. So the fragment of a sentence that says appositional modifier, inhibitor, AMOD, which is an example of which would be galdanomycin, an HSP inhibitor, is able to blah, blah, blah. That dependency path between a gene and a drug occurs almost 1,200 times in abstracts in PubMed. Similarly, the gene activity was insensitive to this drug, a potent inhibitor of cytosolic NQ01. So that has this dependency path. We see it 450 times. So even though there are many ways to say things, they are countable. And this can go on, and I won't go on and on, but this is the top 10 repeated templates. So here's a little test matrix that we made. On the rows, we have pairs of drugs and genes. So the first row is ciprofloxacin and CYP3A4. I'm not even labeling the columns, but the columns are these shared dependency graphs. That is to say, sentence types that we've observed. So there's one sentence we've seen relating ciprofloxacin and 3A4 in whatever this corpus was that we were looking at. The next row is quinidine and CYP3A4, which has two different sentences. So there's no overlap between ciprofloxacin and CYP3A4 and quinidine. However, the third guy, who I'm not labeling, has a sentence that looks similar to a sentence with ciprofloxacin and another sentence, this is the third row here, that looks similar to the quinidine. That third drug-gene pair kind of unifies these three to say they may have a similar relationship, and we just haven't seen certain sentences in certain cases. And then therefore they may form a cluster where the columns are all the different ways to say the same relationship, and the rows are the genes and the drugs that have that relationship. But as you can see, it's filled with holes. It's not a dense matrix. And now I can show you what those sentences are, and they're almost impossible for you to parse, but I've highlighted that we have a cluster here of sentences that are all about inhibition, very similar to the ones I was showing you previously. Inhibition-type sentences. And not surprisingly, the gene-drug pairs that you see in the rows are all inhibition relationship gene-drug pairs. Then there's over here where we see not the word inhibition, but we see metabolized and substrate. And these are all gene-drug pairs that have a substrate or metabolizing relationship. Okay, so obviously I'm not going into the full detail, but I want to show you that you can go into PubMed, you can grab 50,000 genes and drugs that are connected by at least 5 sentences. You can find 197,000 of these dependency paths, which are basically sentence templates, and you can insist that you get at least 5 each of those. And then you get this beautiful matrix of each dot means that a certain sentence has been used to describe a certain gene-drug pair. Now, the magic of this work was that Beth figured out how to smooth this matrix so that she could fill in this square, for example, to recognize that all of these genes and drugs could have all of these sentences, even though we haven't observed them, in the way I was discussing earlier. So she has this amazing set of little boxes. Each little box is a set of genes and drugs that share a relationship. So we said, well, this might be the total space of all gene-drug relationships. So these are just sentence types that co-occur. D is an antibody directed against G. D is a drug, G is a gene. D, drug, is an antibody directed against gene, co-occurs in one of those little matrices with D is an antibody targeting G. Well, that's not a surprise, right? Those, as a human, you know that those are the same sense. But we now have a computer program that has figured that out for all such sentences. One more example. A G inhibitor, such as D, co-occurred at a very high rate with G inhibitors, comma, including D. Again, same sense. Okay, I think you get the idea. So we've grouped together sentences that are saying the same thing. Then we get this, my spectacular Star Wars diagram. What the heck is it? We take all gene-drug pairs and we cluster them together based on how frequently they occur together in that matrix. And so this is a dendrogram. This is a tree that's been just made into a circle instead of a long, flat tree. And it has 25 clusters. So here's cluster number 25, which is the children of this root right here. So everything below there is cluster 25. These clusters represent clusters of genes and drugs that are being described with the same types of sentences. In the outer ring, you see subclusters, which I'll talk about in a second. And then there's other decorations that we'll talk about. But in terms of delivering on my promise, at the high level, we believe that there are 25 different types of relationships between genes and drugs that are mentioned anywhere in English text in PubMed. So, a little bit more. As our validation, we looked at information in the PharmGKB database, which was talking about genes and drugs that shared a relationship that was a pharmacogenomic relationship, usually the gene metabolizing the drug. So there is a density of PharmGKB orange, and I can tell you that that cluster is indeed a metabolizing relationship cluster. DrugBank is a database of the targets of drugs. So what is the protein that the drug binds? And that's blue, and you can see an enrichment of DrugBank here, which is in fact capturing target binding relationships. So this was kind of how we validated that this wasn't all a bunch of mumbo jumbo. And I just want to dive a little bit deeper. So yes, so the PharmGKB cluster in yellow is metabolic and substrate relationships between the gene and the drug. The DrugBank, you won't be surprised to learn, most drugs inhibit their targets. And so we saw a very strong signal for inhibition-type relationships. Interestingly, if you take this inhibition cluster and look at its subclusters, so we have two subclusters, the light blue and the orange. The orange are inhibitors that are antagonists specifically, whereas the light blue are inhibition that's just more general, not otherwise specified. So what you're seeing in the ultrastructure of these clusters are the different subtypes of a general concept. And if I may pause, all of this is kind of for free and done based on a statistical analysis of the co-occurrence of these sentence types. All we did was look at some of the sentences there and say, oh, that's an inhibition type relationship. Oh, look, this is the subset of inhibitions that are antagonists. There's an activation cluster where the drug activates the gene target. That's over here. And all of the sentences associated with it. And of course, activation also has subclusters, traditional agonists, and then ones that stimulate, again, not otherwise specified more in finer grain. So you can spend all day, and we have spent many days analyzing this, but let me just present to you the 25 ways in PubMed that a gene and a drug can interact. Okay? The gene can be involved in synthesizing the drug, the gene can activate the drug, the drug can be a substrate of the gene, etc. Now these are my English language summaries of a rich set of sentences that are contained within each cluster. As you can imagine, we're excited about this because this gives our curators at PharmGKB a closed world. We now can tell them, you are not going to see arbitrary relationships between genes and drugs. 90% of the sentences you see will fall into this category, and therefore you can come up with standards for representing this type of information, and we can build tools to help them more quickly enter information using the knowledge that there's about 25 with some subsets that we can also specify. As you can also imagine, we are incredibly interested in now looking at all the types of relationships that can be had between genes and other genes, or drugs and other drugs. And what I'm most excited about, genes and phenotypes, drugs and phenotypes. And so the lab now is working feverishly to build the equivalent sentence structures, do the analysis, and again, kind of create at least for the first approximation, closed world of the set of relationships that are observed in the literature between different entity types that are of critical interest to pharmacogenomics, at least. And we think, obviously, that this would scale beyond that. You can't read this, but these are predictions that we made. We wanted to win the hearts of the PharmGKB curators because, after all, in some ways, we're trying to put them out of business with the NLP technology. That their jobs are secure for a long time because they're very good. But what we were able to do is give them a long list of things that we found in the literature that looked to us like they could be in PharmGKB, but they weren't. And so the curators took those, did their own independent evaluation, and most of these wound up getting added to PharmGKB. And there's a little bit slightly more readable. So I think 3 stars, let me just go back because it has the 3 stars indicates an association that has been demonstrated experimentally. And then these are the ones we highlighted, and then the PharmGKB guys decided if they wanted to add it. So I'm almost done. In ongoing work, we're trying to scale this up. We're working with my colleague in computer science, Chris Ray, who won a MacArthur Award 2 days ago. I'm so happy I set up this collaboration before he was a celebrity. For learning from genes, drugs, diseases, and building networks of these interactions that we can then use for more discoveries, similar to what I described for the drug-drug interactions. Once you have this formal network of gene-drug phenotype relationships, you can go to town on making inferences to generate hypotheses for your clinical or biological collaborators at all scales. So we're looking at large-scale analysis of all PubMed sentences, and we're increasingly looking at full text. Thanks to PubMed Central, another NLM product, and the PLOS journals, there is a large and growing corpus of publicly available full text where you just get access to even richer information. So to conclude, I hope I've gotten you excited about the potential for natural language processing research. And in our experience, it's been driven best by a specific application. We're supposed to build this database. We want to do it effectively and efficiently. We need data science tools to help us get on top of this literature. The key organizing entities for us are genes, drugs, phenotypes, and it's very potent as a way to extract information about the molecular basis of disease and therapeutics. And we believe that we can make inferences with this extracted knowledge that would be difficult. Think about that paper from 2000 and the paper from 2005, may or may not share common terminology, difficult to integrate. Computers can do it. So I imagine this emerging network of data, the literature, experimental data, population data, at all scales, as represented in the biomedical literature. I think there's unbelievable opportunities for harvesting that to drive our experimental programs with reason and with fully informed by all the work that has gone before, as opposed to less reason and less informed, which is not what's happening, but it could happen. And I just want to, in particular, thank everybody in the lab, in particular Beth Percha, whose PhD defense I basically just gave. And I want to thank you for your attention. This is the PharmGKB team that works very hard putting out the PharmGKB. And here's some papers describing the work that we discussed today. Thank you very much, and thanks to Don and Dr. King. Good. So please go to the mics. I would welcome questions if you have any. I know it was a little bit of a fire hose, so you're sitting there thinking, what's up? Yes. I'll repeat the question. Thank you.
Somebody in the hospital with 13 drugs, another level of complexity is almost impossible to imagine.
Yes. So the question is, what about, and we've all seen this in those of us who are clinically active, a patient on 13 drugs, and I was just talking about pairwise drug interactions. I have some hope there. The reason I have hope is, if I build my network of gene-gene interactions and then gene-drug interactions, I can look at all 13 of those drugs, and I can begin to see, are there areas of human biology that they are all hitting at the same time? And does that focus my attention and my hypotheses about the potential side effects that I would be worried about? So you might imagine 4 drugs, a hypertension drug, maybe a blood pressure drug, I'm sorry, that's the same thing. A hypertension drug, a hyperlipidemia drug, a diabetes drug. They sound different. When you map them to the molecular level, you may see standard pathways that are being tickled by all 3 of those drugs, and you may say, wait a minute, that's a pathway that I know would lead to neuropsychiatric side effects. So now I'm predicting the potential that these 3, 4, 5 drugs working together intersect in an area of biology that allows me to predict side effects. And in one case, for 2 drugs, we've done some work like this. But the polypharmacy issue, I think, is the question for the 21st century, because anybody who has a patient over 60, they're often on 5 to 10 drugs. They have uncharacterized interactions. The FDA has approved them without looking at those interactions, and so it is up to us in the so-called post-marketing pharmacovigilance to figure out what's going on. So I agree. Let me say the upside of that. It is possible that combinations of medications using the same exact reasoning could be efficacious and meritorious for new indications. So if we look at the progress in treatment for tuberculosis, HIV, diabetes, schizophrenia, depression, there's more and more an ethic of polydrug hitting the pathway in multiple places. So we worry about the polypharmacy for the side effects, but the upside is that same polypharmacy may suggest new uses of old drugs in combination to create new therapeutic strategies. So I like to be an optimist. Dr. Florence.
Thank you very much. And I'm still thinking about those curators you were talking about, and the kinds of, given the world that we know is here, the data's flowing, publication models are changing, about the kind of intelligence you imagine adding or new tools that you think they might need or want, or expanding even into that full text world you talked about.
Yeah, so I made a joke about replacing them, but I actually don't think they will be replaced. Most of what we're doing in the R&D portion of the lab is building tools for them to use their domain knowledge to more efficiently triage potential things to add to the database. So we want to bring the sentence up to them. We want to show why we think this sentence is suggesting a relationship between the gene and the drug. If that relationship has been mentioned in 70 other sentences, we want to have a statistical summary of how many of those were negated, how many times did it say no, how many times did it say yes, and then let them make a judgment call. So really this work is in support of curator enhancement and not replacement.
Well, I think it's important because it's hard to see how we can scale if it can't go that route.
I am also a little bit optimistic that as we're enhancing, we will find that there are certain easy cases where they come to me and say, Russ, can you just put those in the database because we're at 100% rate when certain things are true, we're accepting. But I can't tell them that. I need them to come back to me and tell, just for political, social reasons. I want the curators to decide when the tool is good enough so that parts of it can be closed loop automated.
Thank you.
Thank you.
Russ, I had a question for you about the dependence on the literature for this. Here at NIH and probably at every biomedical venue, there is currently a great concern about the reproducibility of a great deal of what's published. And it's not just an occasional bad actor, it's really a very large segment
Yes.
of it. And I wondered how you see that influencing the type of work that you're doing here.
So the issue of reproducibility is a big one, and I would argue, I'm biased, but I would argue that this is the mandatory first step to understanding what's going on, because we can tell you, as I kind of inferred, implied in my answer to Valerie's question, we can actually tell you all the sentences that have related a gene and drug, and we can tell you two things. How many of them said X is not related to Y, or X does not metabolize Y, versus X does metabolize Y. So just because everybody's saying it doesn't mean it's true, but that's a piece of information that's useful. The other thing that we've experimented with, which has been fascinating, is looking at the occurrence of these things over time. So you definitely see certain facts that were true from 1990 to 2000, and then they were not true from 2000 to 2001 because all of the sentences were negated. And so I think the temporal analysis is very interesting and maybe useful for reproducibility. And I think this as a basic infrastructure to identify the important low-level inferences which are clearly problematic and have not been agreed upon, I think that could be the first step in then creating a science of the detection of problems and then the triage of those problems to say, where should society spend its resources clarifying a clearly muddy situation? Yes? I wasn't keeping track, so we'll just go back and forth until we're stopped. Please.
Hi. So thank you for a wonderful presentation. And you seem to have these 25 relations that describe the whole universe. And it might, do you know about the big mechanism where the big mechanism project run by DARPA,
yes,
where they big these incredibly complex models? So how do these two relate?
So I'm not involved in the big mechanism product, but it is a very exciting, it's a DARPA-led initiative to try to do computational thinking on the mechanistic level, which is very compatible with our goals here. I'm a fan of the effort, haven't been involved, and would love it if some of the work we've done here can be thought of as relevant and we could start participating, because we're certainly going to do this anyway, and if it would be helpful to that effort, we'd love to be involved.
So, as you're programming for, novel drug interactions, have you ever thought about bringing that to the molecular level, where you look at functional groups and structures of chemicals, and then try to use your program to infer of new, molecules
Yes,
that might have?
It's a great question. One of the beauties about informatics and data science is you are not tied to scale. So while Beth sits in the lab doing this work, I have other students who are working on the analysis of binding pockets in 3D structure. It's all data science, it's all informatics, as long as you understand what you're talking about, which they do because they learn. So we are very excited about taking molecular level interactions and looking at how they're described in the literature, and then using these to either cross-correlate for increased confidence or to debunk because of irreproducibility type reasons. So yes, and in fact that goes to expression data, genomics. I think it's the most exciting informatics is the integration of these data at different scale to both corroborate or to not corroborate inferences made at one scale.
Thank you.
My college roommate is here today.
So English is usually the language I use when people stop me and ask, what is it you're trying to say? And so for publications in English, there is a limitation. There's probably a limit to how good the parsing can be.
Yes. There were 5 sentences in PubMed that crashed the Stanford parser.
Excellent. Could you see the Stanford parser being used to pre-process abstracts so that a journal will ask, is the abstract saying what you mean it to say? And do you think that over time, journals need to get authors to produce something that is more computer readable, or Should natural language process continue to get better, or is there some way the two technologies can merge?
That's a great question. As a youth, I was very enthusiastic about the power of journal editors to get authors to do things, like enter structured representations of their findings and use ontologies to encode so that I wouldn't have to do any of this. And it might be my particular experience, but I have lost almost all hope that that would ever happen. Biologists are busy and productive and great, experimental biologists, and the chances that when a paper is finally done and accepted that they will now take the time to encode it in a controlled language, unless we have unbelievably fun tools that are fun at the level of video games, which is not out of the question, I don't see that happening. So it's a little bit of a pessimistic thing to say, but for that reason we're just saying, okay, let's assume they're going to be writing natural language, and it's our job to extract that natural language from the text and also from the tables and figures, which actually are a little bit more structured. And there is exciting work, not in our lab, but in other labs, looking at extraction of knowledge from tables. But maybe I'm wrong, and maybe some energetic young person will win a Pioneer Award and try to do this. Structured entry of biological findings. I think this will be the last question.
That's a great slide for my question, which is, what about reuse and other use of your data sets, possibly even commercial use?
So maybe you could qualify a little bit more what you mean. So almost everything we do is put into the public domain because of the NIH grants and the desire to just kind of push it out. So I just want to make sure I understand.
Well, there are other text mining and drug development efforts, some of them commercial.
Right.
Are they using your data? Is your data available
Right.
to them?
Right.
Do you share data?
Right. So yeah, a great question. There is a very rich NLP community, and because of the most recent publication, we're starting to get inquiries, and so that's exciting. The other thing is I'm recently doing a lot of work with the FDA. The FDA has a lot of text, you won't be surprised to learn, and it has a lot of good information in it. And so there are regulatory science applications of some of these technologies that I think would go right to the point of impacting the regulatory process in the hopes of speeding it up and making it more effective. So yes. And I think that's it. So thank you very much for your time and attention. Okay.
I hope you'll stay for just a moment. We have an award, and I'll ask Dr. Lindberg and Dr. King to come up and present the award to Dr. Altman. We will have one for everyone we have in this series as we go along. In addition, in the Lindberg Room at the National Library, we will have a board, a plaque on which we will have, Russ, your name permanently engraved. I also want you to notice that this award is very, very close to being a royal crown, which is what we intended.
Oh my goodness. There must be a photographer. There he is.
It's very heavy.
It is very heavy. Okay. Tell us when to stop. Thank you very much. This is wonderful.
I'll fill that with cookies for you.
Thank you so much. Thank you.
Thank you all for attending. We do have some light refreshments as a reward and a thanks to all of you. They're in the FAES area not far from here, and there are people right outside who will guide you to it if you can spare a few moments from your day job to relax a little bit. And thank you all.